anaerobic infection. So we will brief here. These are component of human the mu- flora, but process spore form bacilli, gram-positive are the Clostridium species, and the non-spore forming gram-positive bacilli are the Actinomyces, Bifidobacterium, Lactobacillus. Where is the Lactobacillus? Commensal lactase. What is the site? Vaginum. Okay. So they maintain the pH. So if there are the less lactobacillus on the slide smear, then there is a probability of infection. Gram-negative, among gram-negative bacilli, there are bacteroids, Pribotella, Porphyromonas, Fusobacterium. These are the example of gram-negative bacilli. These are the anaerobic examples of the bacteria. In cocci, we will see the Peptococcus, Streptococcus, and Veillonella and Megatheria. They are the gram-positive and gram-negative cocci. These are only anaerobes. There are many, but these are most common cause of anaerobic infection. So disease caused by anaerobic bacteria, there can be any infection from mouth, head, neck, thoracic cavity, abdominal cavity, pelvic cavity, skin and soft tissue infection to central nervous presenting on they are present on the depend on the sinus. If there are sinus affected, there is a sinusitis. If there is ear affected, otitis media, that is otitis externa or otitis dental involvement, or there is pus or inflammatory infection. So abscess like liver abscess, lung abscess, ocular infection, any abscess, or any gangrene. Later the stage is gangrene. So exogenous origin or endogenous origin of anaerobic infection, they are divided based on the exogenous or endogenous origin. So exogenous origin caused by the any exogenous cause like food or superficial wound infection, or if there is a associated muscle gangrenes, or any trauma associated with tetanus or cellulitis associated with the infection like benign superficial or deep infection, or there is IV drug. So these are the exogenous cause of anaerobic infection. And anaerobic infection which are the endogenous in origin are the antibiotic associated. What are the antibiotic? You should know. Aspiration pneumonia. What is the aspiration pneumonia? Anyone? Please respond. What is the aspiration pneumonia, or what are the risk factor for aspiration pneumonia? Aspiration of gas any this type of patient on ventilator. Okay, during endoscopy, there is a aspiration of gastric content, so there are aspiration pneumonia, aspiration in lungs of that content, or complication of further any inflammatory condition like appendicitis, cholecystitis, endocarditis. ठीक है, any bone anaerobic, exogenous, endogenous, or infection. So, what are the major clues for anaerobic infection? If there are infection or infection, you found the infection is anaerobic or anaerobic infection. So, in anaerobic infection, there are the mainly site exudating is fallen order, necrotic tissue found at the site of gangrene or exudate on examination. There is a black discoloration or black pigmentation containing exudate around the site or on the site, or there is a presence of sulfur discharge of actinomyces infection, or there is a pills in distress in bite wound, traumatic wound in case of bite wound or gunshot injury or any accidental trauma. So there can be breach in normal flora. So there is a easy to cause the anaerobic infection or secondary to the animal bites or antibiotic. So first you should know the reason, then you what a specimen anyone please probable specimen and suggest one or two specimen. Yes, good. There are many reasons of aspiration, coma, infants, due surgery, old age, immunocompromise, altered consciousness, endotracheal tube insertion. Thank you, good. So what are the probable specimen? What specimen will you suspect? When you suspect the anaerobic infection, what specimen will you collect? Never swab, yeah. Biopsy, base of lesion, any aspirated fluid, स्पेसिमेन कैसे आएगा? वाइड माउथ कंटेनर में या सिरिंज में? Fluid. ठीक है, टैप. Okay. See the next slide. What are the specimen here? So there are blood. First and foremost, blood. Second, fluid. If we suspect the bone tissue aspirate, bile aspirate in case of GIT infection, or endometrial tissue or suction curettage in case of endometritis, bronchial washing, ठीक है, or in case of suspected CNS lesion, if there is no any reason found out like aerobic infection, fungal infection, parasitic infection, then we will see further for the CSF for the anaerobic infection. We will take this sample and then fluid from normally sterile sites like joints, peritoneal fluid, percutaneous fluid. These are the many samples we will collect for the suspected anaerobic case, depending on the site of infection. So there are many samples. So can be used. It should be oxygen-free transport system or food or fecal sample ideally transported at 4 degrees centigrade in case of poisoning in Clostridium perfringens. And serum, stool. So in wound botulism, what are the samples like serum and stool or feces for the Clostridium difficile for culture and toxin assay. And the specimen for toxin assay may be collected in a leak-proof container stored for up to 3 days at a 4 degrees centigrade only. So choice of specimen depend on the site of infection. What are the transport methods? So exposure should be avoided for oxygen. So there is a stored transport media. Like example of stored transport media are the sodium thioglycolate media, sodium glycerophosphate media, calcium chloride or methylene blue or distilled water also, carrier media, anaerobic transport media, Robertson cook meat broth, simplest and easy method, syringe and needle sealed with a sterile rubber stopper or CO2 vials or bacteric anaerobic culture water जो automated वाली होती है. तो these are the transport media if there is a delay between the patient and the lab. So these are the method to transport sample. So this is the bottle, culture bottle, anaerobic culture bottle. So please check the label before the collection of the specimen. The bottle is anaerobic or for an aerobic method. This is the stored transport media, how it looks. And this is the Robertson cooked meat broth. This is the peptone water and these are the cooked meat particle. These are the method to collect the anaerobic specimen. These are the devices, collection devices containing an agar or indicator system, which is free of oxygen, free of CO2, as well as free of N2. You will have in the hospital this kind of devices for collection of sample in suspected anaerobic infective cases. So these are the sterile transport media for specimen, like sterile pack contains a sterile swab and an oxygen-free inner tube. These are the gas pack pouch. We contain if a tissue, if we need to transport a tissue is placed in a small amount of saline or culture plate are inserted into a self-contained atmosphere generating anaerobic bag. These are the gas pack pouches, and these pouches contain the palladium catalyst as an anaerobic indicator. Advantage of this, but we will not open the plate. We can see directly or we can observe directly the plates in this kind of polybag. So early growth of colonies can be readed without opening the plate. So this is the advantage of this gas pipe pouch. But if we need to process that colony or that growth on that plate, so this is the disadvantage. We have to open that plate. So this is the disadvantage of gas pipe pouches. An advantage, we can see only read or we can see the colony. So what are the method for creating the anaerobiasis? Anyone knows, please suggest me or please give me some example of method. Anoxomat, paraffin, yes. Gas pack, yes. Or Macintosh jar, roll tube method, flight jar, glove box. Good. We will see candle jar. Good. So we will see the method. So what are the method here? What we are describing later. See. First, the methods of creating anaerobiasis. What are the methods? So these are the methods. First is the evacuation replacement jar. We will replace the presenting air in that jar. Jar can be metallic or they can be glass jar. Or second is the anaerobic jar with disposable gas generator. Okay, they will create the gas and they will dispose the all same evacuation plus gas generating system. Third is the anaerobic glove box technique. So we will avoid the exposure of the oxygen in this technique. Or roll stick tube with pre-reduced sterilized anaerobically sterilized media. So these are the plates available. We will describe it later. What are the plates? Which are the anaerobically pre-reduced, anaerobically sterilized. We call this media pass media. So conventional anaerobic jar used as a evacuation and replacement of the gases technique, like gases with the hydrogen, nitrogen, carbon dioxide. So all gases replaced with these gases. And gas pack anaerobic system. This is the system with the seal foil sachet with the 2 tablet of citric acid and sodium bicarbonate and sodium borohydride and cobalt chloride. These are the catalyst agent. Any one of these can be used. In Anoxomat, this uses the automated evacuation and replacement system technique. And Anaeropac, this absorb the oxygen. Here, the oxygen is absorbed and the carbon dioxide is generated, but not the hydrogen. So these are the conventional anaerobic jar, gas anaerobic system, Anoxomat, and Aeropac, and Aerocult. Commercially available catalyst-free system, which makes of iron filling in sachet to which water is added, producing oxygen-free, carbon dioxide-rich atmosphere. Brass reduce anaerobically sterilized media where we use the roll stick technique. और anaerobic cabinet हो सकती है, anaerobic glove box हो सकते हैं, और anaerobic building jar हो सकते हैं, जो कि inexpensive होते हैं. What are the anaerobic incubation conditions? So there should be 85% oxygen, 10% hydrogen, 10% carbon dioxide. Or there are indicators, there are incubation conditions. What are the indicators? There are semi-solid indicator, indicator of brewer or Algiers, indicator residue in dye, bacteriological indicator like Clostridium tetani or Bacteroides fragilis. So these are the strict anaerobe if they will not grow in presence of oxygen. Or Pseudomonas aeruginosa, if there is a growth of Pseudomonas aeruginosa, means the anaerobic condition is not maintained. So these tell about the bacteriological, these bacteria tell about the conditions of the aerobic or anaerobic or strict aerobes. So this is what is this jar? What is the name of this jar? Anyone attending or not attending? Macintosh jar, metallic or glass jar? Okay, this is the Macintosh or Flight's anaerobic jar. And this is a jar containing the plates. And guess that is the system where the evacuation process is done. First, the leaking is checked about the jar, that is there any leakage is present, and second is the presence of catalyst. Third is the evacuation, and fourth is the proper condition in this jar, like hydrogen, CO2 and N2 ka ratio. And then after this all formalities, this jar will be inoculated into anaerobic condition, not in aerobic condition. And then further, after 48 hours, we will see the plates if there is a presence of growth or absence of growth, or there is a presence of any strict aerobic growth. Anaerobic was the condition in the not maintained. So presence of anaerobes determine the conditions of aerobe or anaerobic. Also determine the that is maintained or not. This is the so we will do this cap. So this lid should be closed during the functioning or during the activities of this anaerobic. This is the roll tube technique. This instrument prepares the thin agar film into inner wall of the test tube. So this is called as a roll stick technique, and 3 middle test tube can be picked. ठीक है. So इसमें ये जो है ये thin layer of tubes anaerobic agar की होंगी, और the pre-reduced anaerobic जो मीडिया था, this is the sterilized media, pre-reduced anaerobically sterilized media की plates हैं. आइडेंटिफिकेशन कैसे करोगे? After incubation, if there is a presence of, we will gram stain that specimen, and we will culturally characterize like hemolysis present, or if there is any pigmentation. So based on the hemolysis and based on the pigment presentation, we will characterize the colonies. We will characterize the type of bacteria. Biochemical identification say toxin assay, animal pathogenicity testing is an option to write in theory, not in the practical. So we not do this test. Rapid enzyme detection test, or these are the high assay to detect, and HLC Malitov the profile detect the aerobic, or to identify the anaerobic infection or presence of. These are the high level take. So what you will see in gram stain? Same as a gram negative bacteria, but we will not characterize on this gram stain. So these are the positive. These are the only anaerobic infection or anaerobic infection. So you should proceed later. These are the typical fusobacterial meanings. Define the fusiform bacilli. These are the shape of the gram negative taper and bacilli. They can be pleomorphic, rounded end, or a bizarre shape, means not any confirmed shape. What is this bacteria? What is this bacteria? This is written over there. Clostridium. Clostridium perfringens नहीं है, Clostridium. Just say Clostridium. So GLC is a gas liquid chromatographic assay. The content of the cell wall, to identify the content of the cell wall, because we identify any syndrome or any history based on the genome pool. In bacteria, we will identify the gas liquid chromatographic assay. So there are the lipid profiles. So particular bacteria have particular lipid profile. So this GLC is a gas liquid chromatographic assay to detect the lipid profile, cell wall lipid profile of the bacteria. These are the gram stain picture. You can draw that line in this line diagram in your theory paper, like gram positive or gram negative bacteria, straight rod, gram positive, short end-to-end chains, rare presence of subterminal oval or free. Is the this less stain area is the spore. So if this less stain area is in center and so a subterminal end, there is a subterminal spores. So these are the defined on the basis of presence of spore in that bacilli. So these are the terminal or subterminal. Clostridium difficile having the subterminal. So this is terminal spore here. Vilonella. These are the gram negative with gram variable. What are the primary isolation, anaerobic or anaerobes? So anaerobic selective media are the selective or non-selective. So non-selective media are the common is the blood agar. What are the varieties of anaerobic blood agar, like trypticase, soy agar, or yeast extract? They contain the trypticase, soy agar, phytone, sodium chloride, yeast extract, vitamin K1, cysteine, defibrinated, shape or rapid blood. So these are the content of anaerobic blood agar. There are the different content of anaerobic blood agar. And selective media, there are name as a, we know there shortly as a BBE, KVLB. So we should know the name. What is BBE? Bacteroides bile esculin agar, as they inhibit the bile. Selective differential media. So we use the antibiotic here, gentamicin, which inhibit the aerobic organism, or we will use here 20% of bile, which inhibit the most anaerobes. So presence of bile, there is esculin hydrolysis, turn the medium brown. So you should know the bacteroids name suggests for the, this media is used for the Bacteroides fragilis group. Kanamycin, vancomycin, lake, blood agar. This is the other example of media for the gram positive and gram negative bacteria, anaerobic bacteria. So here we use the vancomycin to inhibit other bacteria and to gain the other bacteria. Anaerobic phenyl alcohol blood agar to inhibit the gram negative facultative anaerobes and swarming of the proteus as well. Cycloserine, cefoxidine, fructose agar, CCFD is other name for this media. So BB, what are the media? पहले कौन सी मीडिया बताए थे? इससे पहले कौन कौन सी मीडिया बताएं? BBE, kanamycin, KVLB, yes. Peptone is extract both thioglycolate, both glutamate, both. These are the other example of anaerobic media. Then we use the KVLB media here. The before this kanamycin, vancomycin, lake blood agar. This is the hemolyzed blood we use in this blood agar plate. Here the anaerobic blood agar plate contain the blood only, and this KVLB lake blood means the hemolyzed blood, which when the RBC are hemolyzed, the ingredients of RBCs comes out into the media, and they provide directly the all material to facilitate the growth of desired organism. So when we use the lake blood agar, we have the ingredients of RBC out of the RBC cells. So we have the we directly support the anaerobic bacteria for growth by using the lake blood agar. So this is used in anaerobic identification. So lecithin agar or gelatin agar, mannitol agar, these are the further starch agar. These are the further use for further identification of bacteria. So what are the pigment producing anaerobic bacteria? You should know the pigment producing bacteria. Few example of presumptive identification of anaerobic gram-negative bacilli like Bacteroides fragilis group. Here the vancomycin disc and kanamycin disc and colicin disc will be resistant, and while acetylene hydrolysis positive, indole is variable. In pigmented prevotalized pigmented bacteria, so here the vancomycin, kanamycin are resistant. So there are the presumptive identification made by the use of different kind of disc as well as different kind of ingredients. There are other identification techniques like Vitek anaerobic identification take 2 or Vitek 1. So here we use the growth cell suspension of the bacteria from the pure culture isolate. We use desired growth, the broth, and we emulsify the for identification, allow the determination of anaerobic incubation. And these are the detection methods provided on the computer database system. So identification tables make the easy identification. Data are saved and can be transported or generated online to the patient. So these are the rapid technique. Without any delay, we can have the report. There are anaerobic. These are the anaerobic card, ANI card. These are the name as ANI card for the Vitek anaerobic identification. So they contain the 30 wells or 28 wells or defined substrate or enzymatic. For defined substrate and enzymatic reaction, there are the wells, particular wells which contain the chromogenic substrate, which and 8 modified conventional tests. So these are the cards available for the further identification. So what are the tests available? How many tests are available? According to that, we have the wells and we have the substrate and we have the ingredients. If that bacteria or that substrate among the that 28 among 8 substrates, so we will identify the further that which are the bacteria in this card. So different bacteria are positive for the different type of substrate. These are the other commercially available kits, which are the name as a rapid ANA 2 or rapid ID 32A rapid method other than the white tag. So other methods like gas liquid chromatography, this is not this batch के लिए नहीं है ये. You should know just the chromatography name. What is this gas liquid chromatography? This is the chromatography. Know the name, just the name. Any question, please? अगर कोई question हो तो पूछो.